Sorry, it suddenly went to just the presenter mode. All right. So thank you so much for having me here today. I really appreciate it. I'm going to keep an eye on the clock here, so hopefully we can cover things in 40 minutes. Maria asked me to cover things in a more general way, not just specifically about my research, but talking about the bigger picture of adaptive optics retinal imaging. So I'll start with that, and then for probably about the first portion of the talk, and then the second half, I'll really delve into the work that my lab's been doing and the things that we've been working on. And so, as Maria said, I recently moved to the University of Waterloo, but the work coming out of my lab is all work that was done previously when we were at the University of Rochester. And I felt that I'd focus a little bit on that work, just to give you a taste of what we had been doing and some of the things that we will maybe not be pursuing in the future, but that have led to some of the activities that we're going to continue to do here in Waterloo. So, you know, a lot of you work in vision science, you're familiar with that, but as you think about zooming into the eye, the clinical perspective, that wide field perspective, gives us a lot of information about the blood vessels and the general health of the retina. But when you zoom in with, say, a fundus photo onto a small scale, you really don't get a lot of information or detail about what's happening at the cellular level. Whereas if we can image the retina, and this is a picture of individual rods and cones, the cones being the large circles, the rods being the small circles, we see that if we can understand this at a cellular scale, if we can map what's happening, how these cells might be functioning, how they're functioning in healthy eyes, how they're functioning with aging, how they're functioning with disease, if we can start to understand that, then we have a possibility, even if it's at a small scale, even if it's in portions of the retina, at understanding how disease processes take place. What's the timeline of progression of disease? What's normal function versus what is function when things are going, becoming diseased. And if we can catch disease at an earlier stage, then maybe we have a hope of treating it.
Jennifer, sorry to interrupt you. We have a problem with the sharing. We are not seeing it, so
okay, let me try again. Let's see.
I left the meeting and joined again just to see if it was me that I was already sharing, but it's not the case. So I think it's your somehow. Maybe you can try again and see.
Yeah.
It's working now. Yeah.
Do you see it now? All right.
Sorry about that.
No, that's okay. Thank you for letting me know. Sorry about that, because so we're trying to zoom in, look past the fundus photo, and look into things at a cellular scale. And the idea would be to zoom in beyond the cellular to the cellular scale, so we can understand how things function, what cells are changing in different disease processes or with aging. And then we can start looking at different treatment strategies. As new treatments are being developed, can we understand how the cells are changing, how they're responding at that cellular scale before the longer term outcomes that are often used in clinical trials of vision loss and changes on an acuity chart? Can we understand at a very early stage whether or not a treatment's working, whether or not a treatment has potential of working? If we're trying to do a treatment which would affect the cones, for example, are the cones there? Are they able to be restored? Or are we looking at a retina where the cones have been lost? And so a lot of potential and a lot of vision that we have for what we could do if we could actually tap into at a cellular scale and image this. And as well as just imaging structure, also try to image their function. And so as we probe into the retina, in order to look at the retina in a living eye, obviously we can't take it out and put it in a dish and look at it under a microscope, which would give us beautiful quality images, high resolution, but it only gives us one snapshot in time, and it wouldn't be very good for patients. So we use the optics of the eye to image the back of the retina. So think of the retina as sort of our sample in a microscope. And if you think of the optics of the eye then as the objective lens of the microscope, it's going to allow us to image in and see what the back of the eye looks like. Now, a lot of you are working in vision science, working on different properties, and a lot of you will know that the optics of the eye are actually quite poor. And so if you were ever to go out and buy an objective lens for your microscope that had the optics of the eye, most people would laugh at you. They'd think this is ridiculous. But yet that's what we're working with. So we have this objective lens that is the optics of the eye, and so we need to correct for those optical imperfections. And so ideally, if we had a perfect eye We have a larger pupil is going to have less diffraction. We're going to have a smaller spot. A lot of the imaging systems, not all, but a lot of the imaging systems that we use within the eye are scanning a small spot across the retina. And so the resolution and the size of our system depends highly on the size of that spot that we're scanning on the retina. Of course, typically an eye is highly aberrated. And so we would be dealing with a 3 millimeter, 3.5 millimeter pupil if we wanted to think about commercial systems. They're often focusing into the sweet spot that's matched between diffraction and aberrations. But if we can get to a larger pupil diameter, a higher resolution by going to a 7 millimeter pupil, we need to be able to correct those optical aberrations. And of course, to do that, for the past 20 years, we've been working in adaptive optics. Now, adaptive optics, very simply broken down into sort of its schematics, would be the ability to measure with a wavefront sensor. Can you see my mouse?
Yep.
Okay, thank you. Measure with a wavefront sensor the optical imperfections, the aberrations of the eye, and then to apply a correction to a deformable mirror of some sort to correct for those aberrations. And if you can run this in closed loop, some groups run it in open loop, but we tend to run it in closed loop. Measure, correct, we can correct for the optics of the eye. Now, this is typically done with a Schaff-Hartmann wavefront sensor. In the case of a perfectly flat wavefront, going through the lenslet array onto the CCD camera, you can see that it generates a perfect grid of spots. Of course, when you have the aberrated eye, the shifts of the wavefront slope are causing the spots to shift. And if we can measure the shift of those spots and correct them by controlling the deformable mirror, then we can take an aberrated wavefront onto a shaped deformable mirror and have come off of that a planar wavefront. And so by measuring and correcting, we can correct then both the ingoing light and the outcoming light of the eye, so that what we do is we have the best looking spot on the retina by pre-compensating for the aberration. So actually aberrating the beam going into the eye, so the eye's aberrations correct the spot on the retina. And then on the way back out, we recorrect again in order to actually correct the spot that would be coming back out of the eye and heading into our detector. And so with that, we end up with an aberration correction. On the left is your wavefront aberration. You can see when you run the adaptive optics, it can flatten the wavefront. You can see the point spread function, of course, shrinking down to that small spot. And if you think about then a scanning system imaging on the retina, scanning that small spot on the retina is going to have more intensity focused on a small portion of the retina, giving us much better, much brighter images and a much better resolution. And so once we've run and controlled this control loop, then we can put a detection scheme, and this can then, adaptive optics can be combined with whatever detection scheme we would like, or it can actually also be combined, and I think a lot of people here are probably working in the field, where you combine it with sending light into the eye and actually having visual stimulation on the retina and doing psychophysical testing. We're interested in my group and with the retinal imaging into seeing those cells, putting some sort of detection scheme and retinal imaging system on the back end here. And so a lot of different ways to do that. This is an example just showing a histological cross-section of the retina with all of its different layers. And a commercial SLO system has poor axial and lateral resolution. But if we start to combine adaptive optics with a scanning light ophthalmoscope, a flood illumination system, or even on the bottom here, an AOOCT, we can start to see that the volume of the PSF shrinks down and we can image and get quite good resolution in both of these things. And there's advantages to both using OCT and using SLO, and I'll talk a little bit about those next. So what does this adaptive optics do? You can see here in this video that we go from a blurred area, blurred retina, to when we turn the adaptive optics on, the image gets brighter and we see the resolution of the photoreceptors. And so in AOOCT, again, something similar can happen. Here what you see is particularly in the bottom and the middle, a typical standard OCT image. And then on the left and the right, you can see an OCT image where the adaptive optics has been focused in the outer retina on the left, and on the right, it's focused up in the inner retina. And that allows us to get even the people working in AOCT to get even better resolution within that area. For example, this work coming out from Julien Liu and Dan Hammer have shown that AOCT, when focusing at different layers, allows impeccable resolution. You can see the nerve fiber bundles here on the right in figure E is, you can see the small circles, and the circles sort of in between the blood vessels, which are the ganglion cells. And they're able to start looking now at different soma sizes, compare that, for example, the bottom right is a composite image of the 3 different vascular layers in the retina that they can look at. Of course, with OCT, you can also section in to see the photoreceptor layer as well as the RPE layer. And OCT is a really up-and-coming technique in terms of the AOCT in particular has really boomed in the last several years, and is allowing us to start looking at high resolution both axially and laterally. It's limited though in that it's relying on reflected light and scattered light, and so it doesn't work well with all the things that we might be interested in looking at in the retina. Although there's lots of different functional aspects coming in with OCT, such as optoretinography and things like that right now. Most of what I'm going to show you next, I think the remainder of the talk is really going to be focused on adaptive optic scanning light ophthalmoscopy, a scanning system that images layer by layer rather than the volume based of the OCT. With that, again, for many years, they've been imaging foveal cones and rods and cones. You can see over 10 years ago now that the first images of the rods were shown and with this AOSLO. But with AOSLO, we can do microstimulation, similar to what you do when you do AO psychophysics, but here targeting into sending in a specific letter and stabilizing it on the photoreceptor mosaic. And I show this because not only is it important for the psychophysical testing being done in Austin Rohrder's lab and other groups, but the fact that the eye is moving, we have to compensate for that both in AOCT and AOSLO. And so a lot of effort and a lot of work has been put into tracking the motion of the retina. We can acquire multiple different types of images, track motion in one image, and apply that motion to all the different other image modalities that we may be acquiring at the same time, particularly in an AOSLO. That stabilization, the ability, for example, here shown as the E staying stationary on the same set of cones, can be done both in real time and post-processing. And that stabilization is critical then for generating the images that we're going to see. So anything that you can do in microscopy, I like to think that people doing AOSLO in particular are interested in seeing how we can adapt those techniques to imaging within the retina. And the idea and sort of stimulated, motivated by phase contrast imaging is sort of non-confocal aperture imaging, where rather than using a confocal aperture that we typically use in reflectance imaging, you would offset that and look at multiply scattered light. Examples here show that you can get a much nicer picture of the vascular walls or the cone inner segments, not the cone outer segments as we saw in reflectance, but the cone inner segments. Neural cells such as the ganglion cells and horizontal cells are also visible with this offset aperture, non-confocal aperture type imaging. Jesse Shallick's work at the University of Rochester, monkeys, humans, a variety of things. We can also, though, with this, see improved images of blood flow. Here you see cells flowing through different capillaries. You can see it's non-perfused, reperfusion of this upper vessel here, and then perfusion of this. So you can actually look for those stalls of blood cells and start to see things there. You can also do a line scan modality that allows you to see individual cells flowing through the retina. What's interesting here is you can start to understand that even if vast vessels maybe have the same rate of flow, there may be a different number of cells flowing through them. And a lot of work coming out of Jesse Shallick's lab and others are starting to look at blood flow and information related to that. Jesse's group has also been looking at immune cell imaging, and here you can see different immune cells coming in and out of the retina. This is out of the vasculature in the retina. This was in a mouse where here's a cell leaving the retina. You'll see another one coming back in. And this was a mouse where they induced an immune response, and they looked 48 hours after they created an event that would cause this immune response. So a lot of immune cells infiltrating this part of the retina, but again, very interesting and exciting to start thinking about image with these offset apertures or non-confocal detection modes. Again, things you can do in microscopy motivate the things that we can do in AOFLIO. And so fluorescence imaging, very common with microscopy. So fluorescence imaging has been a major push of my research, and is sort of a lot of what's being done to try to understand function of the retina beyond the blood flow and the immune cell work. And so, for example, the RPE autofluorescence on the left, RPE have lipofuscin within them. It's a byproduct of the process of seeing, and that lipofuscin accumulates with age. And it turns out that some of the molecules within lipofuscin are fluorescent. They tend to sit around the cytoplasm of the cell, near the edges of the cell. And so we end up with what looks like a honeycomb mosaic, where the dark spot in the center is the nucleus, and you see the beautiful fluorescence creating the honeycomb mosaic around the edge. So you can start looking at those cells. This isn't a healthy eye, but if you can imagine how in a diseased eye, this would start to look mismatched, it would start to show larger cells and cells falling out. On the right is a ganglion cell image, and it's in mouse. It's work by Bill Merrigan and Kenny Chong, and they're using a genetically encoded calcium indicator, which has been inserted into the ganglion cells. So either this was done with an AAV virus to put the fluorescence marker within the cells. Lots of different ways to put fluorophores within cells nowadays. The genetically encoded calcium indicator, when calcium flows into the cell, it indicates that the cell is stimulated. When the calcium flows in, the fluorescence intensity increases. And so what you see here in this video is in the upper right, or in the trace along the bottom, is indicating when a light stimulus is being put on. And when the light is being stimulated, you can see that some ganglion cells are getting brighter in fluorescence at different points in time. And if you actually look at these two cells here, you'll see them actually flicker in counterphase. One cell being bright when this light is on, and one cell becoming bright when the light is going off, and sort of that information that we can then gain. So they're doing a lot of work in trying to understand function of ganglion cells based on these genetically encoded calcium indicators. But I was motivated by the idea that are there molecules that are naturally occurring within the retina that we could use to understand retinal fluorescence, and that we could use to understand retinal function. And so we started looking at what molecules within the retina are fluorescent. And we think about fluorescence often in terms of a clinical type of image, autofluorescence, blue autofluorescence, infrared autofluorescence. And those tend to come from the RPE layer with melanin and lipofuscin located predominantly within the RPE. But there's a whole host of other fluorophores throughout the retina. For example, the photoreceptors contain molecules involved in the visual cycle, the retinoids, and several of those are also fluorescent. Macular pigment, it turns out, has some fluorescent properties. Molecules involved in metabolism, NADH and FAD, are fluorescent. They're located within all cells and give us an idea of the metabolism, the energy production within those cells, and how that's functioning. As well as structural molecules such as elastin and collagen are also thought to be fluorescent. So can we tap into some of these or all of these molecules and start to understand how the retina is functioning based on changes in these fluorescent molecules? And so the first target we started to think about was, could we image metabolism and the visual cycle? The visual cycle being the recycling of molecules after the process of absorbing light. So one of the molecules involved in that is retinol. Retinoids, all the retinoids tend to have a very similar excitation spectrum plotted here versus wavelength, as well as you can see the NADH and FAD involved in metabolism. Great, we can excite these molecules below 400, below 450 is good for FAD, or below 500 is good for FAD. But that means we're exciting in the UV, and the UV is challenging for two reasons. One, it's potentially harmful to the retina. We wear sunglasses to avoid allowing UV light into our eye. And two, the ocular transmission window prevents us from actually getting UV light to the retina. And so the cornea and the lens will absorb different ranges of the UV light, protecting our retinas from everyday exposure, but making it difficult to do a direct single photon excitation. So what we can find is if we start to do two-photon excitation, where we use an infrared pulsed light source we can create the same excitations or equivalent excitations as to though we were using the UV. And so with two-photon excitation, that will should safely, being in the red, transmit the optics of the eye, it'll create that excitation. And so we can then collect an emission above 400 or 450, depending on where that shifts. And that will give us good excitation through the of these fluorophores that we're of interest in. We started when I was a postdoc with David Williams in collaboration with Krzysztof Polczewski and Grzina Polczewska. And I've continued to work on this since those collaborations have ended. So two-photon excited fluorescence. Oh, I see that you're seeing this. I don't know how to move it. Right there. All right. So two-photon excited fluorescence ophthalmoscopy. This work is all that I'm going to show you has all been done in monkeys. And here you can see the reflectance image of the photoreceptors, similar to what I've shown before. You can see the large circles are the cones, the small circles are the rods, just to remind you of that. And when we do two-photon excited ophthalmoscopy, you can see that we get a more uniform intensity image from fluorescence. The cones are more filled in, suggesting that the fluorophore is distributed more uniformly within. So on the left, we're looking and relying on the wave guiding properties of light to give us that reflectance image. On the right, we're relying on fluorophores being located within the cells and distributed within the cells to provide the contrast for that image. And so you can see there's no, the fluorescence between the cells, between the rods and between the cones is much less than the fluorescence within the cones. And so we've got that dominant fluorophore coming from within the photoreceptors. We've also imaged other cell layers throughout the retina in the monkey. This was work predominantly done by Robin Sharma for his PhD thesis. And we've looked at the outer nuclear layer. For example, we've looked at blood vessel walls. An artery wall here shows up very brightly with two-photon excited fluorescence. And we can image the ganglion cell layer. You can see that the nuclei don't fluoresce, but that we have fluorescence coming from the cytoplasm and the surrounding area, almost look like little pearls on a string. And we've been able to tap into two-photon fluorescence as in the photoreceptors as a measure of the function of the visual cycle. This is a plot of the autofluorescent signal versus time during exposure to our imaging light. We are unable to avoid the 730 nanometer light actually exciting the retina. So it's a constant light stimulus. But we see that if we start from a dark adapted state and we put the light on, we can see an increase in the fluorescence plotted here. We've applied this to look at models of retinal degeneration and shown that when you have a degeneration of the outer segments, that this flattens and we no longer see this functional response. We've also looked at systemic hypoxia and found that in hypoxia, the rate of change slows during hypoxia and then recovers when animals are brought back to a normal breathing environment. So intensity is one way that we can start to look at retinal function, but we wanted to probe more closely to see what other properties of fluorescence could allow us to understand what's happening within the retina. What are the fluorescence properties of the retina, and how can we understand that in normal, healthy eyes and eventually moving that into diseased eyes? So for example, here we think of a common fluorescence property as being spectral properties, but the challenge is when I showed you those spectra, NADH is very similar to the retinoids, and the retinoids each have, even though they have different properties, they have very, very similar spectra. And so separating spectrally can become very difficult within the retina because of the overlap. But there's another property of fluorescence that is becoming more and more common to consider and to look at. And so if you think about when we send in our excitation, it's a pulse, a train of pulses. And each train of pulses will create some sort of fluorescence excitation, and it will get occasionally a photon emitted. There's a time delay between the excitation pulse and the emitted photon. And if we measure those time delays, then we can create a decay histogram of the number of photons coming back with particular time delays, and that can provide us with information we call fluorescence lifetime. We can analyze this in two different ways. One is to do multi-exponential fitting, where we can create double or triple exponentials. We often work within a double exponential, but here you can see then we can calculate, for example, a mean fluorescence lifetime parameter, which is a weighted lifetime based on the proportions of the different exponential decays that are fit. We can also do Fourier space analysis, where we take the discrete Fourier transform and then plot the sine and cosine components into what we call a phasor plot. In this space, keep in mind that short lifetimes end up further out on the unit on the circle here, and we increase around, we get longer and longer lifetimes, and the longest lifetimes are at the origin. In this case, each pixel in an image can then be broken down into either fluorescence lifetime exponential components, a mean fluorescence lifetime or tau mean, or a single point on a phasor plot. So work by James Feeks and Sarah Walters looked at the fluorescence intensity image on the left and then calculated the fluorescence lifetime, the mean fluorescence lifetime. And when they did this, we found a very clear difference between the rods and the cones. And we found that the cones tended to have a longer fluorescence lifetime than the rods. We'll talk about what this might mean and what these differences might come from closer to the very end of the talk. Another graduate student of mine, Kang Huynh, he then started looking at this in phasor analysis. And when we look in phasor space, we start to see a distribution here. If we separate out the cones, isolate where the cones are located, create a mask where we see the cones here in orange, the rods in green, and color code those pixels, we start to see that, again, the rods and cones separate in phasor space. But we noticed when we were looking in phasor space was that there started to be this lower second cluster, and that lower second cluster didn't correspond to just some random pixels throughout the image, but actually started to correspond to particular individual cones. Anyone familiar with the retina and color vision is going to start instantly and go, well, that's a sparse array of cones, and the color coding I'm using should not hide the idea that the trichromatic cone mosaic has a sparse array of S cones. And so our hypothesis then was that the sparse array of cones, this extra cluster, was corresponding to the S cones. Kang then went on to show that across multiple monkeys with multiple locations imaged, that we could in fact cluster out 3 clusters within the photoreceptor mosaic, looking and seeing the cluster for the cones, the ML cones, the rods, and again, a cluster that we identified as S cones. The proportion of cones that were S cones is consistent with what we'd expect for the monkeys that we were looking at and for their cone identities. With this, we then proceeded to compare, is this repeatable? Are the cones showing up the same? And in fact, if you look here on 2 different days, the fluorescence intensity images, to the phasor plots here where we had 2 clusters and then classified the cones based on their separation into 2 clusters. Looking at the classified cones, we had good comparison between the day 1 and day 2 exposures. Again, if you start looking between these 2 images, you can start to see that the S cone pattern falls out to be the same. We did this across multiple images, and we had good repeatability of these images. We then wanted to go a little bit further to try to understand and confirm that we thought these were S cones. And so we went to the work that Christina Schwartz had done while a postdoc in Rochester before moving to Tübingen, where she selectively damaged the S cones within the retina. And so here you see a swath montaged of monkey retina, and the upper left image, 15 minutes post-exposure to high intensity 7 milliwatts for 2 minutes for our imaging light, tended to result in a darkening of the subset of photoreceptors. Again, we hypothesized that these were S cones. Christina followed these cells up. If you look 3 weeks later, you can still see that there's dark spots. Again, looking 8 weeks later, we continue to see the dark spots. Christina did histology and showed that in similar locations, it was in fact the S cones that were missing from these regions. We didn't have S cones labeled. And although it may look like a control location, 11 weeks later, we can see in the lower right a contiguous mosaic. But if you start to look carefully, you can actually see where there looks like there maybe should have been a cone in this space, possibly a cone should have been here. You can start to see where cones may have dropped out and the rods have come in and filled that space. And in fact, Christina was able to show with longitudinal imaging, that cones had dropped out and that the rods had migrated in to fill the space. A really interesting thing to start thinking about what happens in our own retinas if certain photoreceptors are dying in a sparse pattern, how things should change, how the retina could look contiguous and healthy, even though it's missing, in this case, an entire subset of cones. And so we use this paradigm where we imaged then with a potentially damaging light source the retina. We had previously, weeks before, identified the S-cones. We did it again and identified which photoreceptors would be classified as S-cones based on their location in phasor space. And then we proceeded to follow up with a subsequent image, get the dark cones, and it turns out that 86% of the cones that we classified as S-cones turned dark, compared to less than 1% of the ML cones, reassuring us that the cones that we are identifying as S-cones based on their phasor position are most likely S-cones. I want to tell you one final story, which we are starting to now try to understand. Well, this would be a very expensive cone classification method. It's very invasive, two-photon imaging. You really wouldn't want to use this as your cone classifier. Excuse me. But what it does do is it might tell us something about what's different between these cones, what's different between the ML cones and the S cones. And to do this, we started looking at something called phasor fingerprints, where not only did we collect data in our primates, but we collected data with solutions in cuvettes that would be solutions of known fluorophores. So just to reorient you to the phasor plot, the numbers that you're seeing now would be the sort of standard lifetime values for a single exponential decay. And if we start looking at known fluorophores within cuvettes, we can find where those clusters would be located, for example, for FAD and free NADH involved in metabolism. We can start looking at adding bound NADH. We can't get completely bound NADH in solution, but we can move towards a mostly bound state. And you can see how the shift in lifetime occurs there. Interestingly, this ratio of free to bound NADH is a common parameter looking in research now, looking at cancer and looking at how cells that are with cancer and non-cancerous cells, as well as those undergoing apoptosis, have shifts in the ratio between free and bound NADH. We can look at the molecules involved in the visual cycle. Again, although they are similar spectrally, they have different fluorescence lifetimes, all trans retinal versus all trans retinol. Looking at other molecules of interest in the retina, for example, elastin in those blood vessels. So we can create this sort of phasor fingerprint and start to compare then our data to where things are within phasor space. We can create lines along the free to bound NADH line, and so thinking about how the ratio of free and bound NADH might change in different situations. And again, we can also look at that continuum between all trans retinal and all trans retinol. As you have ratios of both within the cells, you'll see shifts along that line between the two. And so coming back to our photoreceptors, here is the photoreceptor intensity image in the upper left. If we go to the phasor plot, you can see our phasor fingerprint in gray with our fluorescence lifetime data from an example photoreceptor image in color-coded here. Now we've put the color scheme onto the phasor plot, and then that color coding on the phasor plot is used to generate what we're calling the phasor image. And there you can see the rods in teal, the ML cones in orange, and the S cones showing up in purple based on where they were within phasor space. But what we can also see is there's a shift between the rods and the ML cones, and the relative amounts or relative concentrations or contributions to the fluorescence from the retinal and the retinol are different and shifting between the rods and the ML cones. And so we can start to look at how those might change in different situations. The S cones, on the other hand, are not following completely along that continuum. There must be a third fluorophore, something maybe located out here that we have not yet identified. I don't expect it to be elastin and S cones. And so we're looking now to see what fluorophore is out here to try to understand, well, what fluorophore is fluorescing within the S cones that's making them different, making them look different and have these different fluorescent properties. Two other layers that I want to just tap on. For example, we can look at the ganglion cell layer. Here you can see a nice tight cluster of fluorescence from the ganglion cell that shows us that mix of free versus bound NADH within the ganglion cells, and start looking at how that might change. For example, one thing we were interested in is how does that shift with stimulation, or how does that shift with the induction of glaucoma, and could we learn something about cells that are sick? Tapping into just one other example here, if we look at the branch retinal artery and a branch retinal vein in the intensity image, you can see the vessel wall here on the edge. If we color code our phasor space image, you can actually see that the vessel wall shows up very nice and bright in the red, whereas the neural retina shows up in more green. You can see the nerve fiber layer, neural retina, tends to fall along that free bound NADH line, whereas the vessel artery wall is pulling up more towards elastin, suggesting that the predominant fluorophore that we're looking in at these bright vessel walls is actually elastin. And so this is how we can start to understand what's happening, what's changing, and potentially we'd like to move this from looking at this in monkeys to here at Waterloo, we're going to move it into looking at this in mice and start being able to look at transgenic mouse models and models of disease that we were not easily able to do within our monkeys. So hopefully in that brief time, I've been able to convince you that cellular scale imaging has a lot of potential if we can tap into both structural and functional properties. I'm excited about what we've been doing in two-photon excited fluorescence ophthalmoscopy, and hopefully this will allow us to understand what's changing in normalized and diseased eyes, and eventually tap into understanding how treatment strategies might be affecting that within those normal and diseased eyes. We'd also like to excited about the things we're doing with Fluorescence Lifetime. It provides a new dimension that we can look in for functional assessments. As well, we're hoping that we've been starting to port our Fluorescence Lifetime capabilities that we've done with two-photon fluorescence ophthalmoscopy in monkeys into the work we're doing in mice, but also into non-invasive imaging within people, and starting to look at the fluorescence properties of the retina, particularly lipofuscin within the retina of people. So I want to thank my group, great team of people that I was working with at Rochester, continue to have lots of fruitful discussions with them, as well as our funding sources. So thank you.